welcome everybody again to another episode of the ideaXme show, that show that profiles the humans behind the really big ideas that are shaping our world, inspiring future creators. And for all those that love really great stories, I'm Ira Pastor, your health and longevity ambassador. So for the last several shows, we've been spending a lot of time on the so-called different hierarchical levels of the biologic architecture of life, disease, and aging. We've talked about the microbiome, the genome, tissue engineering, systems biology, dabbled a little bit in more exotic areas of digital therapeutics, electroceuticals, and quantum biology. And as exciting as a lot of these areas are, at the end of the day, for them to yield what many of us want, patients, prescribers, payers alike, that is viable therapeutic outcomes in areas of substantial unmet medical need, these drugs, biologics, gene therapy, cell therapies, and so forth ultimately need to be appropriately delivered to target areas of interest, and during their journey maintain their structure and functionality. The general area of drug delivery technologies, estimated at over $100 billion a year nowadays, generally refers to a group of approaches, formulations, technologies, and systems for transporting pharmaceutical products around the body to where they're needed safely to achieve the desired therapeutic effect. So for today's guest, I really could think of no one better to come talk with us for a little while about this topic, take us into the future than clearly the world's leader on the topic, esteemed chemical engineer, scientist, entrepreneur, and inventor, Dr. Robert Langer. Dr. Langer is the David Koch Institute professor at MIT, faculty member of the Harvard-MIT Division of Health Sciences and Technology, and the David Koch Institute for Integrative Cancer Research. Dr. Langer completed his undergraduate studies in chemical engineering at Cornell, obtained his PsyD in chemical engineering at MIT, where he joined as an assistant professor back in 1978, and has since then grown a lab, which is the largest biomedical engineering lab in the world, with over $10 million in annual grants and over 100 researchers. And their work basically is at the epicenter, the interface of biotech and material science, with a major focus on the study and development of polymers to serve as scaffolds for tissue engineering, as well as vehicles for the controlled delivery of drugs, including small molecules, proteins, RNA interference, and so forth. They're also devoted to developing novel medical devices for both diagnosis and therapeutic use, high-throughput screening platforms for new drug development and vaccine design, and in cell engineering via both chemical and genetic tools for cancer immunotherapy and stem cell therapies. Dr. Langer has written over 1,400 articles, over 1,300 issued and pending patents, and his patents have been licensed or sub-licensed to literally hundreds of pharmaceutical, chemical, and biotech companies around the world over the years. Dr. Langer is one of the 5 most cited individuals in history. He's the most cited engineer in history and has received numerous awards for us to get into right here, but he is one of the only 4 individuals ever to receive both the U.S. National Medal of Science and U.S. National Medal of Technology and Innovation. He's also served as both member and chairman of the U.S. FDA's Science Board, which is the FDA's highest advisory board. A lot to go into here, but Dr. Langer, it's an honor, and thank you so much for taking the time to come on the show today.
Oh, it's my pleasure. Thank you.
Absolutely. So in our industry, there's clearly no one that doesn't know about you, but for the layperson that is not that familiar with the biotech space, if you could just take a little bit of time to talk about you, your background, where you grew up, how you got interested in science and health, and ultimately sort of the journey that deposited you at this epicenter of all things happening in terms of material science and biotech in 2019.
Well, I grew up in Albany, New York. I went to the public schools there, and then I went to Cornell to major in chemical engineering, and then to MIT to get my doctorate in chemical engineering. But when I got done, I ended up doing something that was very different for chemical engineers. Most of my friends would go into the petrochemical industry, the oil industry, and I wasn't that excited about it. And it's a long story, but I ended up going to Boston Children's Hospital and Harvard Medical School, and I was the only engineer working in the hospital, but it gave me a lot of ideas. And one of the things that I got very involved in were creating new materials for drug delivery, and that was important to solving some of the problems I was working on, which involved trying to isolate the first substances that could stop blood vessels from growing. Those are called angiogenesis inhibitors. We made some discoveries in the 1970s about how you could alter the structure of different plastics so that you could deliver molecules of any size for almost any period of time. And that got me started. And then I've just kept working on different aspects of that, different delivery projects over time.
Wonderful. It's exciting to see that crossover, that sort of the convergent technology happening, in the sense that, biotech's one of those areas where, in the past there wasn't too much crosstalk, but the fact that you've leveraged it for so many important things. Before we get into some of the stuff that you're working on currently, you're clearly someone that, I guess exemplifies this more than anyone else, but you in many ways sort of make biotech look easy with all that you've done. But most people working within and outside of the space clearly know that biotech is not an easy thing. Very large attrition rates for new drugs, long timelines, dealing with valley of death issues for small companies and so forth. Forbes magazine recently wrote an article about you, citing you as both the Edison of our times and the Edison of medicine. But as we all know nowadays, Edison failed quite a bit on the way to creating the light bulb and so forth. Could you talk just a little bit about sort of the early sort of challenges in your work, sort of getting to this point where it seems like, everything you do sort of has the Midas touch to it now, but in the early days was there, how did you get through that initial issue? I'm sure you encountered a lot of failures on the way, but ultimately that deposited you here.
Sure. Well, first I should say it's not just easy for me now, no matter what gets written. I mean, in fact, I continue to fail a lot, and I think biotech is hard. I think it's very hard. But the great thing about biotech is it can make new drugs, new drug delivery systems, and other therapies and diagnostics that, really can have a major effect on improving people's lives. And that's one of the major reasons why we do it. The other being to do the best science we can. But you're right. Early on, I had enormous number of setbacks. All kinds of things. I mean, first, nobody wanted to hire me for any of the jobs that I wanted. Then, after I did get to make, some findings that I thought were pretty good, nobody would give us any grants. And then I wanted to be a professor, and no chemical engineering department, which was my major, would hire me because they felt like the medical ideas I had didn't make any sense for chemical engineering. And then I ended up going to a nutrition department. But the year after I joined that department, the department head, he was a very good guy, but he didn't ask anybody else in the department whether I should be hired or not. So he hired me, but then the year after I joined, he left. So a lot of the senior faculty told me I should leave too. And so that was all discouraging. And scientifically, the same thing. I mean, you asked me earlier about some of the findings we made on controlled release, but really the first couple hundred times I experimented with different techniques, they didn't work. I mean, so I just kept at it, but it was very tough, very discouraging.
So, nowadays, you're working, in your lab. You have quite a few people on a range of different opportunities. You're involved in the long-acting sustained release side, with traditional drugs, but now there are many more tools out there in terms of the RNA interference, the gene editing, cell therapies. What are some of the things that, looking forward now, the next 10, 20 years, that you're most excited about, that you, obviously everything you're working on has a lot of promise, but in today's, 2019 and looking out, where do you see?
Well, I think there's 3 or 4 things that to me are very exciting. I mean, one are trying to figure out one of the biggest problems with what I would say some of the drugs of the future, and those drugs being gene therapy agents like messenger RNA, short interfering RNAs, different types of gene editing approaches. One of the biggest problems, maybe the biggest, is delivery. People usually say in those fields the 3 biggest problems are delivery, delivery, and delivery. And so we're doing a lot with new kinds of nanotechnology to aid in that. So that would be one area. Another area are non-invasive ways of delivering these and other complex molecules. For example, we recently developed a pill you could swallow that hopefully will enable you to deliver insulin someday. And I think crossing other barriers like the blood-brain barrier, all kinds of barriers end up being very, very important. I think a third area is different types of smart systems,
Mm-hmm.
whether that be ones that you might be able to respond to different signals or ones that you might target to specific cell types, like a cancer cell.
Mm-hmm.
And finally, I guess one of the things that's very exciting and important to me is work that we're doing with the Gates Foundation to try to develop new medical therapies and drug delivery systems for the developing world. And that involves various kinds of challenges to improve patient compliance and create new vaccines and things like that. So those are all areas that are very important to me for the future.
Excellent, excellent. As we move beyond some of the, sort of, I think of that as the, when talks about the RNA interference, the gene editing, and so forth, sort of the cutting edge where we are now. And then, I look sometimes and I see, there's other stuff going on in the industry that I term exotic, where sort of drug companies may be looking to take drug, say, out of the picture entirely. And I point to one of my former employers was GlaxoSmithKline, and they recently invested a tremendous amount of money in so-called electroceuticals, which, for lack of a better word, involves a zap instead of a drug moiety. Also, people in there, digital therapeutics and so forth. Your work, your lab is focused, I mentioned early on, you have some work going on in terms of devices. And at the same time, some of the controlled release systems that you mentioned may involve magnetic stimulation of types, ultrasonic, acoustics and things of this type. Do you sort of foresee a day in the future where there's the drug is taken out of drug delivery, in the sense that there might not be a bioactive moiety that your technology is providing, but once again, the zap or the vibration or whatever it might be independently becomes the drug?
Yeah, well, we are looking at different types of things. I mean, everything from cellular therapies like tissue engineering. We're also looking at new medical devices for a variety of ways. I do think what you said, I think those are all possibilities. I don't think drugs are going to go away, but I think other kinds of things will play a role, and I think that would be great.
Excellent. One area that in recent years that has developed, quite a bit of, legs, I was going to say, is the area of aging. I represent sort of longevity as well as health on this show, and sort of the area of longevity biotechnology has gotten kind of hot in recent years, and there's been a lot going on in the MIT Harvard ecosystem, GDF11 work, Amy Wagers and David St. Clair, of course, for sirtuins, and genetic engineering, and George Church's lab, and so forth. You've been advisor to some of these initiatives. You've also had some your own work in terms of cross-linked polymers, second skin technologies, and so forth. Could you just give me your sort of thoughts, your view on sort of the concept of aging and longevity biotechnology in terms of some of these tools in impacting the future and sort of interfering with processes of aging as opposed to sort of just the outputs of disease?
Yeah, well, I do feel that, I mean, that's a very important area, and particularly if there are ways ever to help people with Alzheimer's disease and, diseases of aging, I think that would be a terrific thing. That's been elusive so far, but I think that would be certainly a wonderful thing and, to see happen someday.
You have Judah Folkman and of angiogenesis fame, and Clark Colton in terms of bioengineering, which you've listed sort of in your bio as important influencers as you moved along throughout your own career. Other people that you might want to mention in terms of those that sort of kept you interested in the space, that had you decided not to do this anymore and gone into something else, got involved in petrochemicals, gotten out of science completely, that you might want to mention?
Sure. Well, I've been very lucky to have a lot of very good friends who have also been my colleagues, people like Alex Klebanoff and Mike Marletta, Nicholas Pepis, and all of them have been, Gray Henry Brem, Jay Vacanti. I mean, so I think all of these people have played a significant role in my life. And then I've been very, very fortunate to have had just wonderful students and staff. I've been very lucky to have a great wife, Laura, and who's also a PhD scientist herself. So I think there are a lot of people that, and my parents, that all played a role.
Wonderful. And now, we typically learn this science fiction question at the end of the show, but obviously a lot of people, when asked, who they want to meet in the industry, they'll mention Robert Langer. For you, if you had the opportunity to meet somebody, now obviously you've met a lot of people over the years and you've worked with them and influenced by them, other people, historic figures, that if you had the opportunity to meet, who would that be for you? It doesn't have to be a scientist, it could be an artist, philosopher, religious figure.
That's a great question. I suppose, I have, in terms of people who are alive, I've had the opportunity to meet a lot of pretty well-known presidents and queens and kings and things like that, and certainly scientists. If I went back in time, I suppose it would be great to be able to meet Abraham Lincoln or George Washington or Thomas Edison, people like that.
I just want to really, thank you. I appreciate, obviously, you're extremely busy with everything you have going in the lab. But it's been a real honor having you spend this time with us and sharing your knowledge. I want to thank you, not just for joining us, but also for the last half a century that you've been developing the modern biotech space, and then obviously everything that's coming in the next 10, 20 years out of your lab's work. Really appreciate everything you're doing and everything and all the ideas and knowledge you're imparting to the next generation. It's really, really great.
Well, thank you for having me, and thank you. Very, very good questions, and I look forward to the show, and thank you for having me.
Absolutely.